Every discovery is the product of multiple minds, sometimes working together, sometimes working in opposition, but incremental discoveries lead to breakthroughs. So the people whom I will mention, that are attributed with discoveries are indeed important people, but there are many others whom I will not mention who participated in these discoveries. So if you want to learn more about the subject, here are some of the sources, ranging from the first chapter of the Vaccines book to a couple of efforts at a relatively restricted view of history, and then a really complete history by my friend Hervé Bazin, which is published both in French and in English. This quotation, which comes from the chapter and the first chapter of the first edition of the Vaccines book, is something which I'm sure no one in this group will disagree with. But just to emphasize that the history that I'm talking about is the history of one of the greatest and most important discoveries in human medicine, that is, how to prevent disease. Well, so let's go back in time to the 18th century, to variolation, which is the beginning, so to speak. Now, where did variolation come from? We don't really know. It has often been attributed to the Chinese, and certainly the Chinese from sometime in the second millennia began to variolate using insufflation, nasal insufflation of smallpox scabs, ground up into a powder. But also, there's some evidence that variolation originated in Central Asia and then spread both west to Turkey, for example, to Africa, and also east to China. So, with that being said, certainly by the 16th century, it was clear that variolation was being practiced in the eastern part of the world. Well, in the 18th century, this lady, Mary Wortley Montagu, the wife of the British ambassador in Turkey, observed variolation and indeed had her son variolated. And she returned to England in 1721, where she began to propagandize for variolation. And eventually, a particular physician, Dr. Maitland, variolated first of all, her daughter, and then others, and eventually even royalty was variolated. But again, Lady Montagu was not the first. Several years before, a physician named Timony, who was also British but who was working in Turkey, wrote back to the Royal Society describing variolation and its value. And as a matter of American pride, if you will, Cotton Mather, the minister in Boston, heard about variolation from his African slave, Onesimus, and he began to variolate about the same time, or even a couple of years before it was started in the UK. So at about that time, early in the 18th century, variolation began to be recognized as a protection against smallpox. Also, as a matter of historical interest, during the American Revolution, when British troops came to the U.S., they had been protected against smallpox. But the American Army had not been. And at a certain point, George Washington had to decide whether to protect his troops from smallpox or not by variolation. Fortunately, he made the decision to go ahead and variolate, and that allowed the American Army to be protected against smallpox outbreaks. Another element that I want to mention is the early discovery of what might truly be called vaccination, and that is, of course, the use of cowpox, or what we now think is probably horsepox. At any rate, although we all think of Edward Jenner as the discoverer of cowpox vaccination, actually a farmer by the name of Benjamin Jesty did the first cowpox vaccination. Now, Jesty was not a physician, and he vaccinated his family, and although that went fairly well, his wife developed an inflammation at the vaccination site, and his neighbors derided him, and so he stopped vaccinating, and thereby left the fame to Edward Jenner, who more than 20 years later began his experiments with cowpox. Incidentally, he vaccinated one person, one girl, and she was successfully protected against smallpox. And he sent this one case history to the Royal Society for publication, and the editor turned him down. So that's an example of one of the earliest mistakes of editors. At any rate, he went back and did more vaccination, and of course became famous for the discovery of vaccination using cowpox. So, moving on from these early days, I wanted to mention that Voltaire, in one of his letters, also wrote about variolation and its value, and this was actually even before he went to England, where he observed it. And this is a picture of Edward Jenner. And this is a copy of one of the illustrations from his first published paper, showing the result of cowpox installation. So then, in the next years, nothing much happened in the way of vaccine development until Louis Pasteur began to think about this. Now, there is a sort of a story that Pasteur accidentally discovered vaccination because a culture of Pasteurella multocida, which causes chicken cholera, was sitting around on his desk during the summer, and when he came back, he realized it was attenuated, and then he had a eureka moment. But in fact, when people have examined his laboratory notebooks, they realized that he had been thinking about the idea of attenuation for some time, and that although it's true that cultures of Pasteurella multocida allowed to sit around would eventually become less virulent, he then went on to experiment with various ways of killing the organism, and then, or partly killing the organism to attenuate it, and thereby to develop a vaccine against chicken cholera. So that was really the first laboratory developed vaccine. But perhaps more important in terms of putting vaccination on the map was the experiment that he did with anthrax vaccine. Anthrax was at the time an enzootic disease in France, and an important veterinary disease. And so his laboratory began to experiment on inactivation of anthrax organisms and spores, using oxygenation, using chemicals to do that. And because there was skepticism about the entire idea of vaccination, an important experiment was done at a place called Pouilly-le-Fort near Paris, where a number of animals, sheep and goats, as I recall, were vaccinated, and a control group was left unvaccinated. And Pasteur invited people to come and see the results of the experiment, and as they did so, the animals unvaccinated were dying, whereas the vaccinated animals were protected. And this, of course, gave him a good deal of fame, but also put vaccination on the map, if you will. So we then come to the story of rabies that everybody knows. Now, rabies, again, was an important disease in France. And the idea of vaccinating against rabies was something that Pasteur was thinking about. He experimented in dogs and showed that he could have some protection by vaccinating using dried spinal cord from rabbits. But a couple of patients actually who had rabies, who were developing rabies, were vaccinated sort of secretly before the famous experience with Meister, the child from Alsace-Lorraine who had been bitten and scratched by a wolf and who was brought to Paris by his mother, and it was decided to try to vaccinate him preventively. And of course, as you know, he survived, and this then became an endeavor of Pasteur to generalize rabies vaccination, which happened relatively quickly. That is to say, the idea of vaccinating against rabies spread rapidly, and as you know, the rabies vaccines are generally quite successful. And of course, his early patients largely survived. This is a photogravure from the Pouilly-le-Fort experiment. Here's a picture of Joseph Meister, who became a guardian at the Pasteur Institute and allegedly committed suicide when the Nazis invaded Paris and a group of German soldiers came to the Pasteur Institute to look at the grave of Pasteur. This was his second vaccination, Jean-Baptiste Goupil, and we move on then to a subsequent important discovery. Now, having had success with inactivating or at least attenuating rabies, the idea of attenuating or killing other organisms rapidly spread. And two laboratories, more or less simultaneously, produced the first data inactivating bacteria. One laboratory was in the US, and the work was done by Daniel Salmon, who also gave the name to Salmonella, and Theobald Smith, a pathologist who worked with salmon. But also a group at the Pasteur Institute in Paris, working in Pasteur's lab, also did early experiments in inactivating bacteria. So a number of scientists then came into the field to try to develop vaccines based on inactivated bacteria. And this is a picture of Wilhelm Kohler from Germany, who worked on typhoid vaccination, inactivating typhoid bacilli. This is Sir Almroth Wright from Britain, who also developed a vaccine against typhoid, which was then eventually used by the British Army in the Boer War in South Africa. Although with great resistance, it is said that on a troop ship, which also carried the vaccine, that the soldiers threw the vaccine off the ship because they did not want to be vaccinated. But eventually, enough studies were done to show that the vaccine was highly effective. This is Emil von Behring, also from Germany, and he was one of the first to show that certain bacteria produced toxins. He and Emil Roux at the Pasteur Institute, again working on certain toxin-producing bacteria, showed that these toxins would induce antitoxins. And this then, of course, became eventually, as I'll tell you, the basis of toxoid vaccines. Kitasato from Japan also took part in these studies in Behring's laboratory. But I also want to mention Paul Ehrlich, who did not develop a vaccine himself, but he was one of the early immunologists who realized that there were substances induced in the blood which could act against organisms, which we, of course, today call antibodies. And he was extremely important in developing the ideas that if you could stimulate the immune system, you could generate protection, at least in terms of serum antibody. So he deserves a good deal of credit for that. Turning to the subject of pertussis, the organism was first cultivated by a Belgian, Jules Bordet, working with the veterinarian Olivier Jambou, so that we have the Bordet-Jambou medium. Before I go to that, let me say that the early pertussis vaccines, there was one developed by a Swede named Thorvald Madsen, but it was not really very effective. And two ladies, and I think I have a picture, let me go to that. These ladies, Kendrick, Eldering, and Gordon, developed the first effective pertussis vaccine. And what they did basically was to grow pertussis bacilli and to kill them. And their vaccine, contrary to the early vaccines, was indeed effective and became the basis for DTP vaccine. So they indeed deserve a good deal of credit. So here is a timeline for the first vaccines. As far as plague is concerned, there the name of Yersin looms important. He came from the Pasteur Institute but worked mostly in Vietnam. And those of you who read French might be interested in a book called Peste et Cholera, a book written recently about the life of Yersin, which is a very interesting book. So moving then to the early part of the 20th century, there was, of course, interest in developing a vaccine against diphtheria and, for that matter, tetanus. And it was known by that time that these were toxin-producing bacteria, but the question was how to work against that toxin. And as I said before, it had become known that there was an antitoxin produced if you use the toxin to inoculate animals. Well, the question basically was, how could you make the toxins safe? And this was done by a worker by the name of Glenny, and Gaston Ramon, working in France, who realized that they could inactivate toxins using chemicals. In the case of Ramon, he used formaldehyde, which has given us the toxin or toxoid vaccines against diphtheria and tetanus. So that indeed the first vaccine certainly licensed in the US, aside from smallpox, was DTP. But also early in the 20th century, there was, of course, interest in developing a vaccine against tuberculosis. And here we see a picture of Albert Calmet, who was essentially the developer of the first, and perhaps the only, vaccine against tuberculosis thus far. Now, incidentally, as you can see from this picture, originally the vaccine was given orally. It was only later that it became an intradermal vaccine. But the point I want to make here is that the vaccine was the result of a tedious process of passage in vitro, something like 230 passages of Mycobacterium bovis, the cow form of tuberculosis. And aside from developing the first vaccine against tuberculosis, which is at least protective of children, the principle of passage in abnormal circumstances to attenuate organisms was really established by Calmet and his colleague Guérin. So this was an important, what shall I say, an important discovery of process, as well as a particular vaccine against tuberculosis. And that became important in relation to yellow fever. Yellow fever was and still is an important disease in Africa, and also it has probably in historic times, recently historic times, moved to South America. At any rate, the first attempts to develop a vaccine against yellow fever were done at Harvard, working with the Pasteur Institute in Senegal. And what they did essentially was to adapt yellow fever virus to mice, to mouse brain. Well, that vaccine was used in Africa for some years. The two people who are credited with developing that vaccine were an American named Sellards and a Frenchman named Legray. But of course, a mouse brain vaccine, as you will realize, had certain risks, and it was very reactogenic. So Max Tyler, born in South Africa, but working at the Rockefeller Institute in New York, tried to adapt yellow fever virus to chick embryo. And indeed, he succeeded. And after passages of the virus in that abnormal situation, developed the 17D strain, which is now the standard yellow fever vaccine strain, and which, as you know, is one of the best vaccines that we have. But again, the principle established by Calmet and Guérin was to passage an organism in an abnormal situation so that the virulence would, for the host, would decrease. So now I turn to the subject of influenza. Now, influenza, again, is a good example of the importance of multiple discoveries. Richard Shope, working in the U.S., was able to isolate and adapt a swine influenza virus in vitro. working first in also in chick embryos. And indeed, even in those days, they realized that the swine influenza was the cause of the 1918 outbreak. But in terms of a human vaccine, it was a group, a British group, that developed the first inactivated influenza vaccine. Wilson Smith, Christopher Andrews, and Patrick Laidlaw. They isolated the virus, the influenza A virus, in 1933 in ferrets, and then went on to develop a vaccine for humans. Now, incidentally, as I just said, they isolated influenza A. It was then later that Thomas McGill, who was one of my teachers, isolated influenza B, and so that gives us the basis for the influenza vaccine that we know today. So here is a timeline for the early part of the 20th century. Diphtheria, tetanus, pertussis, tuberculosis, yellow fever, influenza. And also a typhus vaccine was developed by Harold Cox, then working at the Letterly laboratories in the US. And that actually was an important vaccine in the Second World War, by the way. Well, so that brings us to the time of just after the Second World War. And here we see a picture of John Enders, Fred Robbins, and Thomas Weller. Now, at that time, it was not that there was no cell culture. There were early attempts to isolate viruses in cell culture, but they were sort of gamiches of cells and tissue, and you would inject virus into that gamish, and eventually you would be able to harvest virus. But they had the idea of using explants, that is, taking tissue and putting them into culture. Original efforts, they succeeded first in growing poliovirus and then subsequently other viruses. And this, of course, broke open the field of viral vaccine development, the ability to grow viruses in a situation where you could easily purify them and, if necessary, inactivate or attenuate. So then we move on to the Polio Wars, as I call them. Having shown that polio could be grown in cell culture, it became clear that vaccines could be developed. Here is a picture of Hillary Kaprowski, who was my mentor. I did my, let's say, I did my first viral research in his laboratory in the 1950s. And Kaprowski was an ingenious person, and he had already, when I joined his laboratory, he had been able to grow poliovirus in mouse brain, but of course that was not going to fly as a vaccine eventually. But with the advent of cell culture, he also adapted poliovirus to cell culture and attenuated at least the types 1 and type 3 in cell culture using simply passage and selection of clones of poliovirus. But of course, at the same time, Jonas Salk was also trying to develop a vaccine, but in his case, he was inactivating the virus grown in cell culture, killing it with formaldehyde and injecting it first into animals, showing that he could produce antibody responses that were protective. Salk, by the way, was a relatively quiet individual, although he, of course, became famous. He was not a showboat, so to speak. On the other hand, Albert Sabin, whom I knew well and with whom I argued publicly numerous times, Albert Sabin was a very strong personality. And I mention this because it actually plays a role in what happened eventually. Albert indeed knew exactly what he wanted to do, and one of the important things that he did was to go back. He had been born in Russia, and he went back to Russia in the 50s and persuaded the Russians to do large-scale experiments with oral polio vaccine that he had developed. The Russians indeed did that in vaccinating millions of people, millions of children in particular, and a group of Americans were sent over to look at the results, etc. One of them being Dorothy Horseman, a wonderful person whom I knew well, but unfortunately she was somewhat hoodwinked by the Russians in that, they told her that 10 million people had been vaccinated and none of them turned a hair. And of course, that is unlikely to be true. Even if the vaccine was perfectly safe, there were certainly some reactions. That being said, the oral polio vaccine was licensed relatively soon after the inactivated vaccine. And partly because of the personality of Sabin, the community, the scientific community in general, was convinced that the oral polio vaccine was the best vaccine, and it essentially replaced IPV in the U.S. in short order in the early 60s. Well, having the ability to grow viruses in cell culture led to a number of other important discoveries. This shows John Enders and Sam Katz, who was working in Enders' laboratory. And what they did was to isolate a measles virus from a child by the name of Edmonston, and to passage that virus in cell culture, and again to select strains of measles virus, and eventually to show that the measles virus that they had was attenuated, and that, of course, led to the first measles vaccine. Now, in truth, the first measles vaccine was pretty reactogenic, and indeed was often given together with gamma globulin. But two people went on with that is, took the Edmonton strain and went on to attenuate it further. One being Maurice Hilleman, whom I'll talk about in a moment, and the other Anton Schwarz from Germany. And both of these strains, that is, the further attenuated strains, were then very successful in vaccinating against measles. Here is Maurice Hilleman, who worked at Merck and who was, to say the least, a very successful vaccinologist. He had also a strong personality. The air was often blue when he spoke, but he was extremely important in developing vaccines in the U.S., taking discoveries and bringing them to fruition as licensed vaccines. And to tell a personal story, when several rubella vaccines were developed, including one by Hilleman himself, and there was obviously some competition. One day I received a call from him saying that he had studied the data and that the strain that came from my laboratory was better than his, and he wanted to use it. Now, that is extraordinary in the sense that it cost him or his company a lot of money, but indeed his decisions were based on the science. And so eventually it was his idea also to combine measles, mumps, and rubella. The mumps strain, as you probably know, was isolated from his daughter, Geraldine, so that he had a very personal interest, so to speak, in the MMR vaccine. So here is a timeline for the years after the development of cell culture. Measles, mumps, rubella. Anthrax, it was not a question of cell culture, it was a question of biodefense using the so-called protective antigen, and that vaccine was developed by the U.S. Army laboratories in Frederick, Maryland. Live adenovirus vaccine, also developed for the military, was really the work of Robert Chanik at the National Institutes of Health. Cell culture rabies vaccine goes back to Hilary Kaprowski. Also worked with him on rabies vaccine. And what he did was to adapt rabies virus to cell culture, in particular to human diploid cells. And working with a wide of laboratories, the first cell culture vaccine that could be widely used was developed in Philadelphia at the Wistar Institute. And that vaccine, as you know, not only was safe, not only was it immunogenic, but it enabled one to reduce the vaccine schedule from 21 doses to 3 or 4. So this, of course, was a dramatic advance in vaccination against rabies. Tick-borne encephalitis vaccine was, or several of them actually, were developed in Europe, notably in Austria by a chap by the name of Kunz. And hepatitis B is, again, an interesting story because a number of people, including Saul Krugman in New York, showed that if you took plasma from infected people infected with hepatitis B, and cleaned it up somewhat, make sure that there was no more live virus in it, that you could use that plasma as a vaccine. But of course, that wasn't a very practical vaccine, although, again, Maurice Hilleman had developed it into a vaccine that was sold commercially and which began to have some effect on the incidence of hepatitis. But perhaps of greater importance was the work of the group at Chiron and the University of California, where they were able to insert, through recombination, to insert the gene for hepatitis B surface antigen into cells, in particular Chinese hamster ovary cells, but eventually also in baculovirus. And that, as you know, was the source of the cell culture vaccine against hepatitis B, which has had such a dramatic effect on the incidence of hepatitis. Turning now somewhat backwards to the story of polysaccharides and polysaccharide vaccines. Here again, one has to stress the importance of early discoveries. First of all, let me see if I can. Oswald Avery is a figure who deserved a Nobel Prize but never received one. He made at least two fundamental discoveries, or three. One was he was the first one to show that DNA is the carrier of genetic information. And he has really not been recognized sufficiently for that. But also, he was one of the first to show that bacterial capsular polysaccharides could be used as vaccines. And third, he showed that if you conjugated the polysaccharides with protein, that you could improve the immunogenicity of the polysaccharides. So, but his discoveries were sort of set aside and forgotten until later on. One of the people who capitalized on his discoveries was John Robbins, working with Rachel Schneerson at the National Institutes of Health, and they were one of the first to develop the Haemophilus influenza type B polysaccharide, although the importance of work by Margaret Pittman should be emphasized because she showed that type B was the important cause of bacterial disease, in contrast to the other five capsular polysaccharides, serotypes. So anyway, as I said, Robbins, etc., developed or one of the first to develop the Haemophilus influenza vaccine. Two people working at the Rockefeller, Emil Gottschalk and Malcolm Martenstein, developed the early meningococcal vaccine. Go back for a second. Robert Austrian, working then in New York, but eventually in Philadelphia, developed a pneumococcal polysaccharide vaccine. Now there again, the history is interesting. Almroth Wright, way back in the 20th century, developed the pneumococcal vaccine. It was a whole cell vaccine, but it didn't work very well, so it was abandoned. Then later on in the 20th century, Colin McLeod, who was then at Rockefeller, developed a multiple polysaccharide vaccine. But then antibiotics came along and people thought that we didn't need a vaccine against pneumococci. But Austrian, first of all, showed that pneumococcal disease was still important despite antibiotics, and also developed the first 14-valent polysaccharide vaccine. So he was also one of my teachers. Actually, I did my first scientific paper in his lab. Anyway, the story of conjugation I've already mentioned, Oswald Avery's early discovery, and many people then later capitalized on that to make the conjugate vaccines that we know today. So here's a timeline which includes many of these vaccines. And moving on to the present era, so to speak. This chap, Herbert Boyer, and Stanley Cohen were at a meeting in Hawaii. Now, I didn't know there were delicatessens in Hawaii, but apparently there was, and they met for lunch at a delicatessen in Hawaii during this meeting. And there they conceived the idea of recombination, that is, inserting genes into E. coli and producing proteins through that. And so that led, as you will realize, to a number of new vaccines and the whole idea of using recombination and genetic engineering as the source of those vaccines. Varicella Vaccine was developed in the 1970s by Michiaki Takahashi in Japan. Although curiously, the Varicella Vaccine has been used much more in the U.S. than it ever has been in Japan. Nevertheless, the use of the vaccine has been gradually spreading. You heard today the story of rotavirus, so I'm not going to dwell on that. This is Albert Kapikian, who developed the first vaccine, which, as you heard, was withdrawn. The subsequent vaccines, let me jump for a second here. The subsequent vaccines, first of all, the pentavalent vaccine was developed by Fred Clark and Paul Offit, working in my laboratory in Philadelphia. And the monovalent vaccine was developed by Dick Ward and David Bernstein, working in Cincinnati at the Children's Hospital. So those were the sources of the current rotavirus vaccines. Again, here is the timeline up until 2000, and you can see that I've mentioned most of these vaccines, although it should be pointed out that the cholera vaccine, the one that's used mostly throughout the world, was developed in Sweden, and there's also a live cholera vaccine that was developed in Maryland and Michael Dean's laboratory. And I think I can then move on to the vaccines licensed since 2000. So just to complete the story, the live attenuated influenza vaccine, the vaccine that we use, well, first of all, the first live attenuated vaccines were developed in what was then the Soviet Union by Anatol Smorodintsev. It was used, it has been used there quite a lot, but I think arguably a better vaccine was developed by Humain Massab, working in Michigan, and that was the cold-adapted live attenuated influenza vaccine that we use today. I've talked about the rotavirus vaccines. The meningococcal conjugate vaccines have largely been developed by industry. That is to say, it's hard to put a single name on the development of the capsular polysaccharide conjugate vaccines that are made today by several different laboratories. As far as the human papillomavirus vaccine is concerned, the first discovery that allowed for the development of the vaccine was made in Australia by Ian Fraser. He showed that if you assemble the L1 and L2 proteins of human papillomaviruses, you would make virus-like particles. However, his technique was not very well adapted to industrial production. And so it was these two people at NIH, Douglas Lowey and John Schiller, who developed an L1 only VLP vaccine, which was successfully then transferred to industry to produce the two HPV vaccines, which we use today. And then as far as zoster is concerned, the current zoster vaccine is simply the varicella vaccine made in a high titer, but you may know that the recent publication from Glaxo is suggesting that a vaccine based on varicella glycoprotein will be sufficient to reawake immunity against varicella virus in the elderly. Japanese encephalitis virus has been adapted to Vero cells and is now the vaccine of choice for Japanese encephalitis, although there is an attenuated strain which is widely used in Asia. And meningococcal group B is perhaps the latest vaccine to be licensed. This has been licensed by two companies, Pfizer and Novartis. But the most interesting thing about at least the Novartis vaccine is the use of so-called reverse vaccinology. That is to say, the technique of analyzing the genome of an organism, taking each of the genes and determining whether they're immunogenic, and if so, is the result of the immunogenicity protective, and then selecting the best proteins to go into a vaccine. In this case, 4 different meningococcal group B vaccines plus the other membrane proteins. So this slide simply shows the cumulative number of vaccines developed during the centuries. It shows that there is increasing innovation and that the development of new vaccines is far from over, that this, as I've tried to explain, has been the product of literally hundreds of minds working to produce reagents, if you will, that could be used in humans to protect them against disease. The final result of which is that this kind of tombstone from New England is not, we hope, likely ever to be seen again in modern times. That is, where children die of infectious diseases. So thank you very much. Well, you know, it's too bad that this screen hides this